Hello!
Hi there.
Welcome
Hi
to
there.
Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny. In a
My name
previous
is
life,
Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm Pfizer's and president of Biotech Without Borders, a nonprofit based in NYC, dedicated aim to the public with the
My
tools
name
of
is Faz,
biotech.
and I have a PhD in microbiology, and I've mostly worked on bacteria, but I also work as a research integrity specialist, and I'm currently working as editor for a scientific journal.
every week we meet to talk about
Every week, we
microbiology,
meet to
and
talk
today
about
we're
microbiology, and today we're doing an overview of some of the cool microbiology papers that we've seen this
And
week.
this week we've got the results of our poll for what paper we're going to be diving
Mhm.
deep into, and it's going to be to do with magnets and antivirals. So it's let me look at the title because I
Yeah.
completely spaced on actually researching it. But yeah, it's a paper that's looking on how antivirals work using
See.
using magnets, and we'll talk more about it later when I've got the actual full title at the end of the show.
it's called
It's called
using like some
using
NMR
like some NMR style stuff to see how that um, molecules work.
Yes. And you can follow along with the papers we discuss
Oh, sorry.
this week, and we've on every week actually, because we've got our shared Zotero library linked in the doobly doo below.
and we want to hear from you.
And
So
we want to hear from you. So
so
please
meet us
use the comments or tweet us at
And
microteachsjc.
boy, do we have a show for you today! We've got even though there's scant Omicron news, we have some news coming up on that. We've also got some more
Mhm.
studies into how vaccines work, some new promising vaccine candidates, and a better understanding of how the immune system and what it reacts to during COVID infection. Plus, we've also got some cool bacteria forming structures, so some programmable materials, which is going to be fantastic,
Mhm.
and how things can be used to avoid rust. And we also got have some. What else do we have? Uh, New gene editing like structure that enables us to discover new antibiotics. So yeah, lots and lots of stuff that we'll get into.
absolutely. And I guess
Absolutely.
what's on
And I guess
everyone's
what's on
mind
everyone's mind at first is this Omicron news that we've dug up.
Yes. So
There's been a lot of news out there, but not a lot of
yeah.
published work.
So I've found that like there's been this trend
I've
of like people who've been seeing the same stuff like we do, and they're saying some having an opinion on Omicron, and that's suddenly becoming the news story. Uh, and also like a trend of like because there's a lot of like people who are working like right now, like and and the results aren't there yet. They're still very preliminary. So occasionally like I imagine they get phoned up by a reporter and they just give their like the first thing off the top of their head, and we don't really have concrete data.
Yeah.
So it seems we're getting some
Hmm.
contradictory information. So it's been quite hard to find actual data. But
and
Yeah. And what's this report that you've
oh yes,
the
so now we've
brief
got yeah
from this?
right up now we've got these threat assessment brief. So it's not like this isn't like a proper paper. It's more like a policy document. So it's titled the so it's called the threat assessment brief. It's produced by the European Council. So it's produced by Europe basically. So it's very focused on the European situation, but it does give
That sounds like the CDC
yeah, I
of
think that's the right way
EU.
to think about it. It's the CDC of Europe. Um, So they put this out on to they put out documents like this to give advice to people working in government as to what kind of things people need to do and like giving an idea of what we know about the threat so far and what we need to do in reaction to it.
Hmm.
Um, so there are a few
Hmm.
things about it. So a lot of this is very advisory, but I think the main focus is how much we don't know. So you so,
Yeah.
so for instance, like current estimates of the severity of infection associated with Omicron are very, very uncertain. We it's been like a week or since it's been basically been discovered, and the course of infection for COVID can take 2 weeks or even longer. So we there's lots of data that we just don't have now because we caught this very early. Um,
Right.
uh,
And also, like, to do severity, like, you need a lot of people to
yeah,
be some number of people, right, to get the
significantly different from others,
significantly different from others, and then all those infections are typed, which isn't always the case.
yeah,
you might not get
You might not get
yeah,
sequence.
exactly. So we need more numbers, and but partly because like quite a lot of people are now vaccinated, so sometimes so you have to understand, like, bring that into context as well, as people, whether people are vaccinated or unvaccinated, and checking what the difference is between them. Um, so yeah, and of course, number of confirmed cases on there, and we still have Delta variants. Other countries are struggling with. So I know in the UK we've got like a Delta Plus variant that we're trying to keep. So it's hard to, like, we need to be able to distinguish between both of those in order to properly be able to understand what the difference between Omicron is. Um, so right now I've got.
And then it seems like in this report also summarizes a lot of the stuff that we saw last week and probably what people are saying on the news, right? Like there's all this just like it's the hypotheticals, right, from seeing all this mutation information and trying to trace that back from other data that we already had, right? Like not new data, just like saying like, what if, right? Like what if this mutation existed here? We think that it might have this effect.
Yeah, is the main thing that's changed is that now we've seen it in more countries. So I think when we started, it was like we only really had confirmed cases in like 4 countries, and now it's much more than that. Now it's been spotted at in various places around. This is just a graph showing like the timeline of cases being spotted in Europe. So there's like a big bunch being spotted in the Netherlands and Portugal, and part of this is because it's based on the testing apparatus in those countries. So if the country is very good at testing, then they're going to spot more cases. But I think currently there's 352 confirmed cases as of this report's publication, which was 2 days ago, and across 27 countries. And yeah, 70% of those are from European Union. So, and in some of those cases, we are seeing community and household transmissions. That is, people who are testing positive for this, but haven't been to or from South Africa. So the thought is that it's spreading in the community.
Right. Yeah, so that's not traveler related, right? And this is the thing where if it is an emerging variant, then like this is where it's that community transmission, right, that will drive rates.
Yes. That's right. And yeah, they do talk about what we know so far, like what the sequence reporting is. So you've got like your list of all the mutations, and they try to like make a best guess about immunity, because on paper it looks like there is some questions about whether this could be resistant to antibody detection or other things. Transmission again, it's very early days, but based on what has been seen in South Africa, it seems to have been beating Delta in that area in South Africa. But again, we don't. Again, the area in sub-Saharan Africa is quite a different. Every geography is slightly unique. So I mean, the Beta variant took hold in sub-Saharan Africa and didn't really take hold anywhere else. So, but I mean.
Yeah. So this. I'm gonna say we just got a comment asking, like, I think this is the question on everyone's minds, right? Like, you got vaccinated, so like, are your vaccine titers gonna work on the variant, right? I think that's what is gripping everybody. That's like
that is a big
question, and I think what we're
thank you, Jackie Millender, for commenting. So personally, I think that it will help. So I've been noticing there have been reports coming from Israel saying that we shouldn't be worried about the Omicron variant, but Israel has a lot of vaccinations, so that's why I'm kind of for unvaccinated. So at the moment, I don't think I've seen any evidence that says that we should be worried that
we're going to see more breakthroughs.
Yeah, I think the main thing is not so much breakthroughs, but also like the severe infections, the actual life-threatening infections. There's nothing that say that it'll cause those more often. I mean, it might just be be like a cold. It might. So we don't. But the problem is we don't know, and that's the hard thing that because
I think that's what people are grappling with right now. It's just like we know that there's this potential for danger, but we don't have the solid evidence to say like it is.
My gut says that the 2 vaccines are going to be fine, but I also have this thing in my back of my mind going, I'm going to eat those words if I say them and put my. So I'm going to lay those words out on a plate for myself and go, okay, right, I think that you're going to have a much better chance with 2 vaccines in you. maybe better with 3 as well, depending on how long that time has been from your last vaccine.
I mean, that's the policy. I think it comes up in this document as well, right? Like this document is for government decision makers, right? To in order to influence policy. At the end, they say what's the important thing to do, like non-pharmaceutical inventions, getting your vaccines, right, and contact tracing. So it's like in some ways, yes, like it's business as usual in this sense, but maybe it should highlight to people that like we've talked about this before, the Swiss cheese model or whatever, right? Like, what does it really mean to people on the living their regular lives, right? And not like diving into the intricacies of the data. Well, it just means we should continue to keep up as many barriers between us and transmission of the virus
Yeah,
as
I'm
possible.
I'm getting flashbacks to when we were talking about when Delta came around, because Delta also there was this concern that it might also be resistant to vaccinations. So, and even back before then, there was some worry with the beta variant, and even before then with the alpha variant. So, uh, so there is a part of me that is thinking, feeling like the boy who cried wolf now with like the panic over variants. But this one does seem a lot more different though.
Mm-hmm.
So I'm I mean I'm quite happy to have like because I've had my 2 vaccinations and I think and also I think masking is going to be quite helpful as well to give you that extra level of security. Every
Yeah.
little helps. Um,
Yeah, every little thing helps. Yeah. Yeah, and of course, I think vaccines definitely there's immunity from there. It's just the question is, yeah, what what is it going to get? And actually, we have papers coming up later where we talk a lot more about immunity, especially from the Pfizer one, which I
so
thought was
oh
quite
poor Jackie
interesting.
Melinda. So I've seen her comment. She got coughed on in 2020 with the UK strain and didn't get sick, and then coughed on with the India variant and didn't get sick. Um, so
Uh-huh.
I mean that's
Yeah, but those are like, first of all, that's like an
yeah.
individual's experience, right? It's kind of different from, it's different from like knowing what would happen like in this like
Hmm.
large scale, right? Like, because this document that they're presenting and a lot of the discussion around Omicron, like it's about like what choices should countries make on a whole, right, in order
Yeah.
to
So yeah,
dissuade
this document is very
things.
much
Yeah.
aimed at governments, but in terms of individual, like, can you, if someone has coughed and you didn't get sick, can you say that you are immune? And I'm not sure that you can. I mean, unless you've been to a lab and you've like had your blood test taken, and someone has has basically decided to write a paper on you, so saying that we need to replicate this person's immune system.
Yeah, like a case
Yeah,
study.
so
Yeah. Uh-huh.
I wouldn't take that risk because I've also been coughed on during this outbreak at various points, and I didn't get sick, but then again I've also run through traffic and not get hit by a car. I'm probably not going to stop doing that. So it's
Yeah. Yeah. It's a good way of thinking about what
yeah,
risks are, right? Like just because it doesn't happen doesn't mean that the risk wasn't
yeah.
there.
I mean it's personal choice. All I can do is like say what I would do in this situation. I can't. People have different lives, so I don't know
Yeah.
what I can say for them.
Right. Okay. Do you want to say anything
Ah,
else about this particular
let's see.
document, or do you
Because I
want to
think
look at the
they
next one?
try to. Yeah, basically, there's not really much more because it has just been a week, so we don't know much about Omicron. Um, and yeah, so I mean, yeah, we're probably going to find out more about this in maybe 2 weeks or something like that, because of the way the virus does take time to cause illness. So people are starting to get ill, and from what I'm hearing, like they're not getting severely ill. But that's because it's very early on.
Hmm. Okay. Let's look at the next one. So the next paper is a
Yes,
preprint that
this
we
is a
have.
preprint that has been hitting the news a fair bit. Increased risk of
Hmm.
SARS-CoV-2 reinfection associated with the emergence of the Omicron variant in South Africa.
So in this one, these are the people doing the genomic
Yes.
surveillance in South Africa, and it's kind of funny when I found this preprint. There was an earlier version in November, right? And in November they have this story about how they're trying to model. I think they're trying to maybe they're trying to
Mm.
understand delta spread or whatever. They're trying to understand spread as it relates to like increased transmission rates, right? And so like they have this sort of they have an assumption, right, that um, they have a model and it assumes like a certain transmission rate based on the data from earlier waves, and then they sort of extend that to whatever current case, like they extend that to the data in the present, like whatever we're curious about. So right now we're curious about this Omicron window, right? So like November to December, and they say, well, does our model capture what we're seeing here? Like give it, like they fix the transmission rate essentially, and they say, does our model capture what we're seeing in terms of the new rates? And if it doesn't, then they bring the hypothesis in that that's because something's changing, right, in the virus that's out there doing this particular study.
This study looks a lot at reinfections, not just during, but they look at reinfections during the first, like every wave of this outbreak, right? And they try to like isolate like who's getting reinfected quite often. Yeah, they do. Yeah, this is a lot. There's a lot of modeling here, and there's a lot of. So they take a lot of. So they don't actually confirm whether the people have Omicron in this. They just.
No, it's more temporal. Like they have these parameters that they've seen for reinfection, transmissibility, and reinfection through the different waves, and then they train their eyes on this time period where we think, right, we're hypothesizing that Omicron is driving the bump in cases.
Yes, that's right, and they look specifically at reinfection. So I think they look they class a reinfection as if someone who's like had an infection confirmed 90 days prior, at least 90 days prior to, or I can't, but to the current infection. They have a window, yeah.
It's a window. Yeah, they have a window, and they say, yeah, if it comes up in that window, that might be a reinfection case, right? Like it's not unlikely that that's the same infection that lasted all that time. And then I think maybe what they do is like it's like a little bit later in one of these figures. Oh, I guess figure 3 is like all that data that they have. You can see all the different reinfection events that they map out. So they have everyone, like the first infections are in circles, then people with the suspected second infections are in triangles, and then you got you got squares for the third infection and even fourth infection potentially with these little crosses.
Yes, they've got figure
this is
3.
Figure 3.
Because they noticed that some people who've got like not just reinfected once but twice or 3 times, and that's been quite an interesting thing. So it's an interesting thing to put into context with Omicron, because I think they're spotting a lot more. But another thing that I'd like to think about is HIV co-infections, because in this area of sub-Saharan Africa, that is a lot more common. So that might be something that'd be interesting to like have some information on, to see how much of that coincides with people being reinfected.
I'm not sure if they have that covariate to try to remove from their model, right? Like that could be some information that they can overlay onto these dots.
I don't think they do, because I think they did a lot of data anonymization to protect the identity of the participants. So they only took so when they set out to do this study, I think they only decided to take out like certain bits of information so that they could analyze. So
And
no,
sorry, go ahead.
You got a point to bring up.
Right. Oh yeah, no, I just want to say, like, at the end of the day, I guess it's figure 5. Yeah, I guess it's figure 5 that they're looking at. It's like they're trying to estimate that risk of reinfection and say like increases in the risk of reinfection means something's changing. And so I think the idea is that green bar is the reinfections, and it's sort of going up in this November period. So like something's going on, right? Like this is why like they're saying like this could be the Omicron signal. But it's also like not clear that it's the Omicron signal, right? Like it's only from this temporal understanding that like we think the variant is.
I mean, I'd point out that like in 2020 there's a bump in reinfections, but that seems to be like just from the very first wave. So there are some. This is modeling data, so there is some tweaks. So this is a best guess based on some, but I think the underlying data is somewhat compelling. That they.
Yeah, I think it is. I think it definitely says that, like, uh, especially for this particular, I think because this is like South African data, I would say like this is strong evidence for South Africa to be very careful, right, about I would say like around pharmaceutical interventions and things like this, increasing vaccination rates, and this is like but um, but yeah, exactly what it says about
Yeah.
Omicron is harder
So,
to
yeah,
pinpoint.
reinfections weren't confirmed by sequencing. Vaccination status of the individuals weren't unknown, as was the HIV status. Um, so it is very much people
Hmm.
looking into this data that was already collected without any idea about looking for Omicron, and then trying to look at those trends and then mapping them onto
Yeah.
Omicron. Um,
Yes, yes. I think that that's a good way of summarizing. That's a good way of
so,
summarizing
I
what this
mean,
paper is about.
yeah, in terms of
Um,
extrapolating it to our experience, I think what this the dreaded term this requires more research, but
oh
that's
my gosh, yes.
but that research is happening right now. I think that research started the moment someone discovered this new variant, and there are people who are in their labs right now, like pipetting like the wind to try and get these answers. Oh,
I was just going to say, I'm not really qualified to give medical advice.
yeah, no, I think yeah, need to clarify. We are scientists. We're not doctors. Um,
I'm- yeah, but also like we're scientists who are like reading this for our own interests and not necessarily, um, yeah, not necessarily here to like direct people's actions. It's like more for this curiosity aspect, right? And trying to parse apart like what we think is going on based on the data. But in terms of how that drives individual behaviors and things like that, like you got to talk to your
yeah,
doctor.
uh, for for sure.
All
Um,
right. Um, okay, but along the lines of getting many vaccines, I think that this paper is interesting. This
yep,
BNT162b, that's the Pfizer vaccine, induces B cell responses to SARS-CoV-2 S1 and
yeah,
S2. Um, so what's interesting here is, and I think I might have said this earlier when we got when we first knew that the big vaccine that's going to hit us are these mRNA, like lipid covered vaccine, lipid nanoparticle vaccines. The big question is like, what is it really doing? Because in the advertising decks, advertising, I don't want to say that, but like in the decks that were made by the companies that sort of explain how they thought the vaccine worked, the idea is that, oh, maybe antigen producing cells are going to be, they'll get fused by these little lipid nanoparticles. They will make the spike, right? And then
hmm,
that spike will be processed inside of the very cells that need to process the spike. And that goes on to, touch B cells and all this sort of stuff. But like, there's nothing to say that other cell types also don't get like transduced by this lipid nanoparticle, and so it's sort of hard to know exactly what is going on. And so in this particular paper, they wanted to get some clear data on specifically B cell responses to the vaccine. And so they
hmm,
go real deep in the sense that they pull out the B cells and they look at which B cells, they look at all these different markers on the B cell surface, right? And specifically the B cells that recognize the SARS-CoV-2 antigen that we put into the vaccine. And they get this image. It's almost like, it's
yeah,
single cell, right? The figure one. They're looking at all the B cells, right? And you can see that as the days go by, so
yeah,
like they're color coded nicely, right? In A, you can see the different days in which they've picked up B cells. And you can see how when you spread them out with this multidimensional reduction technique, they're all over the place, right? The B cells are changing from all these different days between but like after the first dose they're changing right into a form. After the second dose they're changing into another form. And then memory as well, like after like a long time later, right, they settle down into their memory phenotype. So this is like a really cool data set and story that follows the development of these different types of B cells. Um, and I don't know what is the clearest figure to point people to that summarizes the big takeaway from this paper, but the takeaway that I was reading as I was going through this paper is something is happening that isn't just more germinal center training of the B cells.
yes,
So what they find a bunch of these memory B cells after vaccination, but they don't have like the somatic hypermutation that would uh, that would say that they've gone through the germinal center. And we talked about the germinal
Yeah.
center before, right? It's like, Faz, I think your metaphor,
Oh
right,
right,
was some
yeah, a
dojo,
dojo
right?
where
It's like
like
it's the training
where
center.
B cells and the immune system train each other to get better at fighting against
Right.
viruses.
Yeah, and here they're sort of reporting an expansion of, of like at that booster dose or boosted dose, whatever the second dose. After, there's an expansion of a B cell population that maybe didn't have to go through that static hypermutation. So it's kind of interesting, like another way to get to memory. Definitely, I don't know so much about like this whole developmental cycle of B cells, and I feel like there's a lot of details here that are speaking specifically to
Yeah.
certain things.
So like the class switching of B
Yeah.
cells and looking, so it really delves deep into like when that switching happens and what it happens to, because they separate, because spike protein, there's like two parts to it. There's the S1, which we talk a lot about because that's got the receptor binding domain, and then there's a second part of it that has the fusion domain that like fuses with a cell. And we often talk about S1 because that's the one where we target all our antibodies to stop from binding to ACE2. But S2 is an important part of it as well, and that also so it looks like there is a response to that. Um,
Yeah. And it's kind of like, I mean, it's hard to say like what is even driving that, right? Like they are using the lens in B cells, right? That's the big contribution of this paper, is that they're taking a very careful eye on how those how those B cell populations are changing over time. But really, like, what is the vaccine actually doing to other cells around it that could be influencing this pathway, right? Because the immune system is a whole bunch of different cells all working in to figure out the best path to sort of produce the immune response, right? And a vaccine goes in there and it dangles these antigens in front of different cells, and presumably, right, like that dangling is going to create a productive immune response, right? Like that is what's showing us the efficacy. And in this particular instance, it does seem like it almost like it's the type of immunity that you're going to get different, right? Regular infection, because for whatever reason, I don't know, but that B cell signature is not something that you would expect, right? From
yeah,
from just like getting infected from like protein based, right? Like type adjuvant
yeah.
style
I mean, it's
vaccine.
interesting how how they seem to find that it reacts to the S2 like earlier than it does to the receptor binding domain. Like in the process of when the immune system
Yeah.
is learning what to react to. I think that's quite an interesting like way to insight on how the vaccine is actually communicating with the immune system to tell the immune system what to be aware of. And I've never really seen anything go into this much detail about that.
I think it actually shows us in some ways, like, and again, I'm just that cogent in like parsing B cell data, but I think it is also helping us understand like that there are many different ways in which you can evolve a B cell response. And if you think of the vaccine, how it indiscriminately
Mm.
fuses to cells, right, and supplies a little bit of mRNA, right, spike mRNA, like who knows, like what cell, right, that received that spike mRNA displayed it in a certain way that is triggering this type of response, right? So I'm sure that MHC restriction type, right, is going to play into
Mm.
this in some way, right? Like why the first one is S2 and the second one is
Well,
S1, something like that. Like there must be plays more likely presented in one version
yeah,
or
because
another.
I presume with S2 that when the virus is like when the spike's like gone into the cells, there is maybe some bit of S2 hanging out on the surface maybe, and then when spike is when
Hmm.
like the previous like kind of spike particles. So I guess maybe there's something in the timing of infection, but again, it's quite hard to figure out, and that's quite an interesting like question to think about.
Yeah, and it's not infection, right? Like this is
Yeah,
this is the vaccine doing this, right? Like this is the vaccine, like in like making our cells make the spike
yeah,
protein, and how our body decides to respond to that, because it's a very like that's not something that just like happens, right? Like this is a technological
yeah.
intervention, like forcing our cells to make this protein so that it would create a good immune response, and it's doing it in a way that maybe we're learning something. I mean, maybe
Mm.
people already know, right? Like the mechanisms that allow for this type of maturation to happen, but maybe this is also a place that we
Yeah,
can
I
learn.
mean, I'd love to see like what the comparison would be between this and an actual genuine coronavirus infection to see what the differences are, because what we want to do, yeah,
Yeah,
the idea of
exactly.
that,
Or like this and like an adjuvanted, right, protein extracellular, right? Like, is there something about like the cell that is being targeted, right, that is changing the profile
yeah.
here? Or yeah.
I mean, what can we do to like communicate with the immune system to make it produce the best response to SARS-CoV-2? And I think this paper does kind of
Right.
edge into the question in a different way by looking at how we're communicating it at the moment, and then from that we can build onto
Yes.
that.
Yes.
Wow.
Yeah, that's what I think is interesting about this hypothesis on how the vaccine actually works mechanistically, right? Not just that like broad level thing. We make the spike and then we become immune to it, but like what cells are being targeted, what pathways, right? How do those cells develop? And that's what this paper
Okay.
is about.
So on the subject of vaccines, we've got another paper called "A single-dose BCG adjuvanted COVID-19 vaccine provides sterilizing immunity against SARS-CoV-2 infection." So
Yeah, this one is. I mean,
Yeah. Yeah, this one is, I mean, BCG, we've talked about this before, right? It's a tuberculosis
yep,
vaccine. And it was shown early in the pandemic that, and I known even before that, that having a BCG vaccination sort of makes you, for a brief amount of time, resistant to a bunch of illnesses. So I think they identified it first in the flu. But then they were like, oh, it actually might be up for COVID too. And there were clinical trials that went in to try to describe this particular phenomena. And in this paper, the authors are like, well, while that might increase like the baseline, right, of immunity that is striking folks, it won't produce like long-term memory because it's just like the BCG vaccine is just like riling up your immune system to be like, let's make sure we fight off stuff. Like it's thought to be innate, the innate side. And so here they combine the BCG vaccine with like a typical SARS-CoV-2 vaccine strategy where they give you some spike protein and adjuvant and they see in mice, and they see really good
yeah, this
results.
like they really give a lot of adjuvant. So like BCG is almost thought of as an adjuvant in itself, and then they add
Yes,
like
yeah.
the COVID. They got the trimeric spike protein they added in the mix, and then they also add alum as well. So the yeah,
Yep.
they test like almost three different flavors of vaccine. One with the BCG adjuvant, one with the alum adjuvant, and then BCG Covat, which has all of them. And then they compare them. Um,
Yes.
so
Yes.
and it really is the BCG
And it really is the
Covax.
BCG Kovacs. So I guess in E you can see, or 1E, sorry, yeah. In 1E you can see that the BCG, right, just by itself, like the spike in the BCG is producing like a
yeah,
bunch of these T cells, right, that are activated, right? They're making interferons, so like there are active T cells. But yeah, you get a whole bunch more when you do the
yeah,
Kovacs, which is all of it, right? Alum and it. So like it looks like BCG is better than alum by itself as well. So right, like there's this. It's interesting to see, right, like the efficacy, I guess, of the BCG adjuvant in the
yeah.
system.
And I think the thing about this that they want to bring across is that BCG is already like approved in humans along with coronavirus vaccines. It's like so I think the thinking was that this could become active a lot quicker than say something that's been tried out that's never been tried out before. Um,
Yeah.
but yeah,
I just want to just pause for a second because I saw a comment come up in the chat just talking about like how much booster we'll need, right? And this is part of that discussion, right? We've talked about B cells in these last two papers. B cells being the cells that are making antibodies, they eventually settle down into a state where they're memory, right? So having like a little bit of the target antigen is going to cause them to proliferate really fast, right? And then we're going to get a whole bunch of antibodies, right, very quickly for that. And it's unknown, right? I think that that's another open question that we're trying to grapple with. And also a good point in the second paper, we don't really know like when did people get their vaccines, right? So how does waning immunity impact, right, increases in reinfection? That's a separate question, right? That sort of demands a separate analysis, and we haven't seen that in these past
yeah.
few papers.
So I mean, I think we've delved into waning of vaccines previously, and I think that there might be something there. But again, with coronavirus we're dealing with the unknown, so there's a lot of precautionary principle style decision making happening where people are preparing for the worst, and so that's why they're encouraging booster vaccines, because there isn't much harm from vaccines, but if immunity wanes, then that could cause issues. Um,
Right. Right. Um, and so I just want to get us to figure two to see D, E, and F, which I think are also really nice. This is like another way of looking at that immune response, right? Not just activated T cells, but like where are those B cells, right? Like, do you have
yeah,
lots of active B cells, spike-specific B cells, spike-specific helper T cells? And here, the BCG plus alum plus trimeric, it really goes to town in terms of increasing these metrics.
yeah,
And so again, this is like, it's kind of interesting because like the T cell metric here, like I'm not sure how that one necessarily is going to relate to neutralizing
yeah,
antibody. And so, right, like there's these other elements, right, of immunity. Like I think from what I read in that threat assessment, people do want to see the neutralizing antibody titer from specific variants, right, on in individuals, because that has that because of all the clinical trials that we've had and experienced, like it's been like the de facto
yeah.
correlate of immunity.
It's a lot harder
for
to get
folks.
like T cells as a correlate for immunity, because there's a lot of complexities there that
Yeah.
I think we might get into a little bit. But antibodies are a lot quicker for people to study and look at, but they aren't the whole of immunity, I think. I mean,
Yeah. And even and in this paper here, right, like we're seeing that like there are other metrics that we could look at to say a successful response in mice.
Yeah.
Again, we're talking about mice here, but you know, it's hard to say, right, without all that data being collected on a larger scale, like what correlates might exist out there for a productive for our protection. What are the
yeah.
human
And
correlates
yeah.
besides neutralizing antibodies?
Speaking about mice, I think the thing that, I mean, they are very different from humans. They use like a special type of mice that is vulnerable to coronavirus infection, because usually they're not. This has got ACE2, so that means that usually it'd have like a very bad infection, and when mice get sick, when they start feeling sick, they stop eating, so you see like a loss of weight. Whereas like when they get the BCG Kovac, you actually see like they're gaining weight, which means they're very happy, which is kind of a good sign. But then again, I know that BCG itself, because I had the BCG when I was a kid, and I've still, I think, still got a vaccine scar from that. So I know that some people might be upset about that. I'm not, but you know,
but it complicates things too. I mean, this is like BCG. It's not really given in the States, and it's because like they want to be able to use like a skin prick test or whatever to see if you've been TB positive. And if you've had BCG, you can have like a cross reaction to that. So it like inhibits. Someone made this decision. I don't know when this decision was made, right? But someone made this public health decision to be like, we don't give BCG. Right, typically, because maybe the risk for tuberculosis is low and they want to be able to surveil for it. So I mean, again, like what's the probability that we'll see this particular, you know, BCG Kovacs mix, or Kovacs BCG alum spike trimeric spike mixed vaccine come into humans? I'm not
yeah.
sure.
I feel like the vaccine market has kind of been cornered by the people who were like there first. So it's gonna be, so unless it has like a significant like benefit, and one of the benefits could be the protection against tuberculosis, because that's still a disease that's around.
Yeah. But I think what is exciting to me always, I mean, this is a sort of like a more like of a very specific case of using BCG as an adjuvant, but like maybe there's a component in BCG, or like if we understand how that tickles the immune response in the way that it does, then we could get to better adjuvants just in general, right? Like maybe this can like guide some sort of adjuvant design.
I mean the idea of like turning, because I think the idea has been around of like turning BCG into like a living adjuvant by like, because it's a living bacterium, and if you can figure out how to like get it to express, I think it's a mix of bacteria.
Is it a lot?
Oh, it's heat killed. All right.
I think it's heat
my
killed.
mistake. Okay.
I think it's
yeah.
heat killed.
Whoops.
Yeah, I think it's heat killed.
Don't quote me on that. Don't ask me for medical advice.
But yeah, but I mean, like again, like there's adjuvants I think we talked about this, right? When we were talking about the R21 adjuvant, you're very excited about this cool new adjuvant being used for
Yes.
malaria that also ended up having like really powerful effects in other disease models. But yeah, like BCG
Yeah.
as an adjuvant, right? Like people who are researching that, like these slight formulations you can make, and like this is going to be a much more stable vaccine than the
Yeah.
lipid nanoparticle one as well. So yeah, there's just all these different. I mean, technology is super complicated, and I love when we're finding like new information about the things that we already use, right? Because it just those technological objects, I guess, are just much more in our historical knowledge, and we have a better we can digest them maybe easier. But also just maybe showing that we can use things and we still don't 100% understand them, but like it
Hmm.
works and it works, right? Like if you have the right efficacy outcomes attached to the way that we do things, like in some ways, people don't dig deeper into like why exactly. And like that's sort
Hmm.
of the curse of science, right? Like every question you answer just opens up all these new questions about like what we don't know about how something goes. And you don't really know like what little tidbit of knowledge is help inspire, right, a new line of investigation.
Okay. Yeah. Keep staying on the like idea of the immune.
A long rant.
Sorry.
Oh no, I just commented on
Oh yeah.
myself ranting about.
Yeah. Speaking on like the immune system, I found this paper called titled an immunodominant NP105 to 113 B star O 07 colon O 02 cytotoxic T cell response controls viral replication and is associated with less severe COVID-19 disease. So this is part of my search for like the perfect T cell response to SARS-CoV-2. So I think I've been on a bit of a tear recently looking for
Uh-huh.
papers that show this. I think 2 weeks ago we had this one on like put on T cells that recognize like polymerase are often very good at clearing out SARS. This one is is a much more well studied candidate. This one, so the first part indicates a SARS-CoV-2 protein that it reacts to, so nuclear protein. So this is like an epitope. So this is a fraction of the SARS-CoV-2 that is recognized by the T cells, and
Really small fraction of the amino acids, right?
yeah,
105 to 113. So it's like just that eight eight amino acid
yeah,
chunk.
this has been found in a number of studies to be quite an important one, especially in combination with B star 10702 HLA type. So, what is an HLA type? HLAs are these major. They're also the same as matrix compatibility. They're proteins that hang around in your cell. They pick up random cells, usually part of protein degradation products, and they present them to the immune system. So they're basically like snitches to the immune system. A very important part of them, and they talk specifically to T cells. And the thing is, everybody has different versions of these proteins. So that's why people have different HLA types. So some people might be more susceptible to a disease, and some people might be more resistant to disease. Sometimes, and the HLA types play a
They're
role in that.
clamps. I think I was gonna say it's kind of nice to think about these as like they're clamps, right? They clamp onto the and so there's this huge diversity because you know what is between the more diversity there, just the greater sense, right? That like you be able to cover every single proteinaceous object that you would want to clamp in nature, right? Like will be covered by the diversity of all the different HLA types.
Yeah, and so they found this combination of HLA type is very good at spotting this nuclear protein. So they wanted to study that in a bit more detail. So they took a look at a sample of individuals who'd like recovered from COVID-19 infection, and looking at and looking at the type of infections they had. So whether they had more severe infections or mild infections, and looking at how well that's reflected by the T cells they have, and what if they can spot this epitope, whether that had a deciding factor. Um, so
Yes.
they kind of like hone in on that for a bit in this paper. So they look and they find that this like tends to be seen in more mild infections. So the idea being that because these people had this T cell response, they ended up having a much more milder infection. Um,
Hmm.
and yeah, so
Yeah.
sorry.
That's actually it's actually very exciting, right? Because the signature of severe disease is one of those things that if we knew better about what that signature is, we could better prepare to yeah we could better prepare like to identify those people that might go down that path. And so actually one of the implications of this could be that if you don't have this type, you could go down the
Yeah,
similar path.
and I think one of the implications is also that if we can understand how this process works, we can design better vaccines and better treatments to essentially tell the immune system how to find the virus.
Yeah. Yeah, and actually it reminded me when I was browsing through this paper, it reminded me of something that you brought up last news period, where you talked about a vaccine that uses a nanobody to direct a specific MHC
Yeah,
response.
but
So I think it was like a nanobody that actually
yeah,
bound an MHC and then brought the spike protein to those cells.
yeah,
Yeah.
I was thinking about that as well, and like the idea between that was directly targeting like a cell type. But if if you're able to like create a protein candidate that can fit into that does the same thing as this does, but also fits into like multiple HLA types, and that'd be very useful. They also looked at like the T cells that bound this as well. So because again, the T cell receptor doesn't just bind it binds the HLA type as well as the the peptide the epitope from the virus. Oh, today I'm losing my words. I'm sorry.
I was thinking about like I know that we read last week was about cell type specificity, but like target something to those cells because those would be the cell types that you would want to have a specific MHC with a specific epitope load. And it would be like the way you get to that. I'm not sure. That's like something happening. I haven't read this paper or when I skim this paper.
Yeah, so I think this is quite kind of a complex question because of the variability, and you don't want a vaccine that only affects a certain small amount of the population. So so understanding how this process works could be quite useful for understanding how it might be applied to other epitopes that maybe don't have that kind of recognition from the T cell response. So
Yeah, well, I guess like maybe part of the reason I'm thinking this way is sort of like it's like from the tetramers thought, right? Like tetramers are just like they're research equipment, right? Like they bind a specific right B cell receptors and T cell receptors bind them, and then they just have whatever is attached to them, some glowing molecule. But like if we made or whatever that's embedded with the specific HLA, right? That's presenting the specific epitope that all has like the stimulatory factor on it to the immune system to do a thing, then like that's the thing that goes in. So, and that the immune system in a certain way.
yeah, and another thing I'd like to point out is that understand this SARS is part of this paper, but I think another big part of it is trying to understand the T cell response as well, and using SARS as that kind of as that model organism because this can be applied. This kind of logic can be applied to other viruses as well, like.
Right, that there might be a specific epitope on a specific HLA that drives good.
Yeah, I mean, we get people who are elite controllers who don't get infected, with a certain disease, and understanding how their immune response works is really helpful in understanding how we can protect everyone.
Yeah, totally. Yeah, I mean, I think that like I always think elite controllers in the subdomain of HIV research, right? And like people have looked at those individuals to look at what antibodies they make. But what about this question, right? What specific epitopes do they display on their NHCs, right? Yeah, it might be part of the puzzle.
This kind of illustrates why it's so difficult to study T cells because we have this problem with the HLA types, the fact that it's diverse between people. So it makes it a bit that it adds an extra level of difficulty to studying them. In addition to the fact that you have to isolate the T cells themselves in order to find out whether they're responding, and that takes a lot more effort as well.
Right. Yeah, but really great find.
Yeah. Next paper, dual function monoclonal antibodies VIR-7831 and VIR-7832 demonstrate potent in vitro and in vivo activity against SARS-CoV-2. So I have a convention to make. I was clickbaited into reading this one because Pfizer sent out this press release saying we've got a two monoclonal antibodies that can react to SARS-CoV-2 spike with mutations significant to Omicron. And in my head, I read, oh, it can react to Omicron, but no, they were very clever with the way they phrased it, and I didn't
Yeah, mutations. If I did Omicron.
So yeah, so what they did was this one is so they got two antibodies here. They're both isolated from original recipe SARS. So SARS back in 2003, SARS-CoV-1 we now refer to it, I guess. Um,
Yeah.
well, I'm the only person who's
Classic
ever referred
SARS.
to it that. Don't follow my example. So yeah, they derived these from a parent antibody isolated from the memory B cells of people with SARS-CoV-2 who'd survived it. And these the thing about these antibodies is that they target a glycan that is attached to the spike. So the spike protein has like a bunch of like sugar molecules that surround it, and this one targets one of those. And thought was that this is very evolutionarily conserved, so therefore it's more likely to hit a broader spate of variants. Um, so they did a bunch of binding assays, and I was very excited about this paper. I was like, oh my gosh, they've finally done something with Omicron. And what they did was remember how like last week we were talking about all the mutations that were significant for Omicron, so things that had been seen before in other viruses. During the course of this research, they were looking at all the variants to see whether their antibodies could work against them, and so they took that data and they put it together, and they put all the mutations that were found in previous strains that were found in Omicron, and they put them together, and they put up a profile saying like, oh, our antibody works against all these when these spike have these mutations. But again, it doesn't say.
And sorry, did they engineer SARS-CoV-2 pseudovirus?
Yeah, they engineered a panel of SARS-CoV-2 pseudoviruses, each with like mutations that were found in previous variants, and they tested their antibodies against them. And so, because they had this panel...
Yeah, so it's very post hoc that they add Omicron into this. It's just like, Omicron has a bunch of these mutations, so here's some data, right?
So, like, look at all these mutations that Omicron has. Our antibody can work against them, but that's not the same as saying it works against Omicron, because Omicron has a bunch of other unknown mutations as well. And, again, until you put it into a petri dish and test it yourself, like, it's very hard to extrapolate. So, yeah.
But it is sort of like, I mean, we've been talking about these monoclonal antibodies, and just recently I was like, someone at a workplace was telling me that, like, oh, like one of their family members got sick and they were treated with monoclonal antibodies. So, like, these monoclonals are really coming into the clinical setting, and like research and development on them is producing these therapeutics. I think they're expensive therapeutics, but they're producing some therapeutics.
I mean, I think that one of the biggest fears for like the Omicron is to do with the monoclonals, because they're a lot more vulnerable to these sorts of changes. I think most of the studies we found I think a lot of the studies that found like immune evasion come from people studying monoclonal antibodies and looking at whether whether the spike can evolve away from them. But then on the other hand, a lot of what happens in a petri dish might be different. I mean, there's a lot of things.
Yeah, I mean, it definitely is different than what happens, and like saying like neutralizing ability, but like there's still the idea of like how quickly does it replicate, right? How well do these mutations let something bind to the receptor, right? That's supposed to be part of the transmissibility question, and like looking at just neutralizing by monoclonals doesn't scratch the surface of the significance of mutations in those arrangements.
I feel like monoclonals they affect the specific monoclonals, and I think people who are giving those monoclonals will know that. I think it's been widely publicized that the mutations are in this region in areas that might. So they might. I mean, there are loads of molecules that are still being produced which affect different parts of the spike. Because
it might be removing tools from a physician's toolbox, but the physician probably has a bunch of toolboxes. Will also know what's going on in this instance. But I mean, now this is kind of like a weird segue to like the paper that we'll cover, like in a month or whatever. But like that's why people are excited about antiretrovirals, because like it's something that potentially like they don't involve monoclonal therapeutics. Like a different therapeutic that targets something critical that isn't changing.
Yeah, I mean, a lot of the antivirals target the polymerase, and viruses can't change that very easily without changing like their survivability. So
but
and
cool. It's good to know that they're still doing monoclonal research, and always
so I mean, this this treatment looks like it could be promising against like some of the mutations associated with Omicron, but again, I feel like I was tricked by it a little bit. So, but yeah, I think that brings us to the end of
All
the
right.
SARS-CoV-2 portion of our show.
But we still have 3 other papers that are non-SARS-CoV-2
Yeah.
related. And the first one, I believe you found this one, was gene editing enables rapid engineering of complex antibiotic assembly
Yes,
lines.
but this is one that I'm quite interested in because, again, antibiotics, we're running out of them, and we always need to be able to create more. And one of some of the most powerful antibiotics we have are ones that are non-ribosomal peptides. So that is peptides that can't be created by ribosomes; they have to be created by other complex chemical reactions. And it's very hard to.
Yeah.
And usually they're multi-step.
Still enzymes. Enzymes make them.
Yeah.
Enzymes make them.
Yeah. You can make them in in labs with chemistry, but the chemistry behind them is very complicated and very expensive to do.
Oh.
So in this,
I think that field of chemistry that seeks to make these types of things called total synthesis. I've heard that before, where it's like you choose like a really complicated structure. You say like, how do I get there? Just using raw chemistry. But evolution has been wonderful for creating these molecules because like genes essentially and ribosomal.
yeah. And I think there's some research into looking at non-ribosomal peptide factories, essentially, like ways to create these. I think we did one of the craziest papers I saw in last year was this one paper where they did like some analyses on some genomes they found in like just in the environment and decided to predict how those genes would turn into enzymes and thus predict the synthetic pathway that these non-ribosomal proteins would do based on just the genetic code alone. It looked like incredible. But this is somewhat related to that. This seems like a bit more like practical in the. So what they did was they like.
The idea that these.
Sorry.
Go ahead.
Yeah. They.
Go ahead. Sorry.
So they decided to like focus in on trying to take a known pathway and then evolve it to become something different. But you can't just evolve randomly. You have to target specific areas. So because evolution can. It doesn't have a mind of its own, so things can go wrong. So they wanted to focus on an area that they know wouldn't mess up the whole enzyme. So they use CRISPR gene editing to do this. So CRISPR can do that kind of targeted evolution by doing like various substitutions and exchanges of different almost like thinking of like taking a Lego set, breaking off one piece and putting in another, like in terms of the active site.
Yeah. No, I think that's a really good way of thinking about it. Like, it struck me that these are already sort of modular machineries that exist inside of cells to create these molecules. And in some ways, this is like re-engineering them and like tweaking them for our own optimization so that they can become factories for all sorts of different complicated molecules. Like, look at that molecule, that's a monster.
My eyes are bleeding slightly from looking at it. It's a very complicated thing, and so in order to put that together, they have a very, very involved enzyme. So I think the main domain they were looking at was this thing called a flavodoxin-like subdomain, which does a lot of the grunt work on this, and swapping different ones. So they know a couple of different pathways for this, and the idea was if they swapped like domains between different pathways, you could get different reactions and different outcomes. So I think they focus on this thing called endurocidin, which is produced by Streptomyces fungicidis, and it's a product that's very complicated, and it's very similar to ramiplanin, which is another very promising antibiotic, but it's because it's got such a complicated structure, it is very difficult to synthesize. And ramiplanin is quite one of those antibodies that are promising against like vancomycin-resistant Enterococcus. So there's quite a lot of interest in finding a quicker way to assemble them. So relatively, endurocidin is relatively lower complexity, so it's a good way to start when you just want to start out on a new technique. Start off simple, walk before you run, and that's what they did here. So they worked on the endurocidin pathway to do some swaps of these flavodoxin Lego pieces, I guess I'm going to refer to them because I keep forgetting. The FSD regions is what they call them. Swap them between them in order to see how
Uh-huh.
that changes the structure of their product.
Yeah. This is cool
Yeah.
because it
This is cool because it almost is like they don't really know how the change is going to
Hmm.
affect the structure, right? Like they just do it to get this idea of like what is flexible, right? In this evolved, right? Because the cluster is evolved to make this specific endurocidin, but like we're trying to now pick apart that all those evolutionary steps, mix and match them to see if like, oh, actually, can this be this evolved pathway? Can it be a scaffolding to put on our own designs and have molecules that we want made by this cluster of proteins of enzymes?
Yeah, I mean, I love the kind of way they approach it is very much from an engineering perspective by the looks of it. Just the thinking of like
Hmm.
what can we do to get the best output, and what are the potentials, like what like testing this way of engineering this chemical pathway to produce a useful antibiotic. And it the
Hmm.
thing
Hmm.
that I was thinking of is like how much this increases the potential for creating new antibiotic candidates or even new just drug candidates that might not have been possible through conventional means
Yeah.
or might have been very difficult and expensive through conventional means.
totally, totally.
Totally,
And
totally. Yeah, I actually like it makes sense to me. I didn't know that this was a category of enzymes, non-ribosomal peptide synthetases, but it makes a lot of sense now that I think about it, and also like kind of cool, like a class, probably very divergent in terms of their evolutionary origin, but like this class of things that is they're made their use in the cell is to
Hmm.
create very complicated molecules. So like understanding like what those how those enzymes work in more detail like can lead us to creating our own complicated molecules, right? Using those as like the basis, the
Yeah,
scaffolding.
and even thinking back to like those metagenomic studies where they're going out and finding these various pathways in genomes, but because they can't culture the actual bacteria that produce it or fungi that produce it, they don't know what those things produce. But then using this kind of gene swapping technology means that you've got an opportunity to access those reactions and then to create another diversity.
Yeah, that's true. You could maybe, oh, that's really cool. I didn't think about that, but that's a very cool, that's a nice thought. It's like getting insight into the metabolism and the pathways that are happening in
Yeah,
metagenomic samples through some basic in vitro style reconstitution, right,
yeah.
of their
When often when
pathways.
we look at like like studies that are there to find new antibiotics, so you often see like people testing a panel of
Mhm.
chemicals and seeing which works. But this looks like it's expanding that panel of chemicals that can be tested on. So so that's why it's kind of
Mhm.
interesting for me to think about.
Cool. Okay.
Ooh, next one. This was quite an
Let's
exciting
see.
one actually.
What's the next one we got?
programmable
programmable programmable microbial ink for 3D printing of living materials produced from genetically engineered protein nanofibers. Yeah, so I guess this is an area that I've heard of it in the background, 3D bioprinting or microbial ink. I guess it can be used in a whole bunch of different ways. They don't actually talk about too much of the potential applications, but the takeaway idea is that, there's all sorts of weird biofilm sticky stuff, right, that's made in the microbial world. Why can't we print it into the shapes that we want? And furthermore, why can't we then engineer the organisms that are in it to do specific functions, to make different things, to turn on circuits and stuff like that? And so in this paper, they look at curly. It's like a protein layer, protein nanofiber
Hmm.
that exists in E. coli, and they engineer it as this ink essentially. So they I guess they engineer some different binding capabilities on
Yes,
it.
they
Um,
they strengthen the extracellular matrix of the biofilm. So I think they essentially using this to
mhm.
manipulate the way biofilms are created. I think a little bit.
they're like printing a
They're like
Yeah.
printing a biofilm, right? Like E. coli uses this stuff in its biofilm to adhere it into the matrix, but here they like make a bunch of it and they mix it with the E. coli to essentially print out a biofilm of E. coli. So you get the shape that you want, and I think that that's one of the big things that they were talking about is that um, I guess like some other technologies have different difficulties in embedding organisms into matrix, like maybe they swell or
Hmm.
like they're too slimy or like something like that. But I guess the physical properties, this is like the next two papers we have are very like
Yeah.
materials focused, right? Like this is like some engineer saying like the physical properties of this biofilm are very nice for printing.
But yeah, I think that there's a lot of like really cool things in there, in the way that you can use this to segregate communities of bacteria, or like so they.
Yeah. Okay. So wait, in Figure 3, can we go down to Figure 3G? I think this is like to me this is the
Figure
summation of how the
3G.
engineers do
Okay,
their
that's
things, right? Like they, yeah, right. Like they show that like when they put these two curly eye, this biofilm protein stuff all together, right? The A plus the gamma, they'll bind together and they'll make this strong sort of material that can bridge these
yeah.
large gaps, right? So like they're very focused on like the structural element of these proteins, but the extension of the technology, as you were saying, Fuzz, is like you can then put
Yeah.
bacteria in these things, right? Like you can embed it your favorite engineered bacteria.
Yeah, and we do have some nice pictures of like some 3D printed like bioink, I think.
Biofilm, yeah, bioink.
Yeah, like
Yeah, they got these shapes. It's a 3D printer that has a syringe attached to it. That's how they're depositing this stuff.
yeah, and so you've got this material,
And
the
in figure,
material
oh yeah,
that
go
I
ahead.
think they describe as like bound nanofibers that display knob and hole domains that kind of link up together to form like nanofibers, and then they can arrange them in
Hmm.
like using supramolecular cross-linking. I guess that's like molecular cross-linking, but like a higher level structure. Um,
Yeah, I think it's like if they're talking about printing. They're talking about they can squeeze it out of a they can squeeze it out of a small hole and it will bind to the things
yeah,
around it, right? It's like glue. They're like kind of spooging out this like biofilm
yeah, and
glue.
then they did like a study to like where they put in E. coli into that glue, and that E. coli was programmed to like
Yes.
detoxify some bisphenol A. I think is a yeah,
Yes. Yeah. Yeah, BPA, the stuff that like you don't want in your Nalgene bottles, your plastic water bottles. Yeah, they also make the E. coli make a
yeah,
dye, a zurin, and then they also have like a switch that turns on and off the growth of the E. coli. So this is where I'm sort of they don't really go into the details of exactly what applications they imagine here because like I would wonder in each of these cases like where would this be useful versus just releasing the bacteria in general right into these locations. And I so it's like very
hmm,
fundamental building blocks, right? Like they just found this great matrix and they can show that in that matrix the bacteria are alive, they're stuck in the matrix and they can do their circuit functions. So it gives this control. What it will be used for, I
I
don't know.
guess
I'm not really
if you want
sure.
slightly different environments for the bacteria, you'll need to like keep them separate. So if you've got like two bacteria, one of them's anaerobic, one isn't, and you want them to be somewhat communicating with each other, then you can print these like kind of fibers in like together, or you could or like I guess there's some
Yeah.
like
Ooh, what about like for like interesting co-culture techniques? Like you want to design like a bioreactor made of two different bacteria, right? Like we were talking about before, the lysis
yeah,
and the cross feeding, right? Like one group is supposed to do one step, but the other group is supposed to do another step. This is a way like you can really modulate the physical characteristics of how much surface area is being exposed for the two different groups of bacteria in like
yeah.
some bioreactor. I'm imagining like some bubble tea style bioreactor, right? All these like little goopy things floating around, but like those goopy things are like optimized for some surface area ratio that increases the production in the
Yeah,
bioreactor.
I mean, I guess the thing is, like, this is one of those things where we don't know. It's an interesting technology, and someone's going to come up with a lot of imagination. Will be like, okay, we're going to make a shirt out of this that's alive, and they can turn your body odor into biofuel or something. Uh, don't quote me on that. That's probably not going to happen.
Yeah, patches. Oh yeah, who knows how sticky this object is? It could be for patches. You could have little
I mean,
patches
I
or
can imagine
something.
like lots of bio art like expressions because we are seeing lots of bio artists come out and like create things like a shirt that can sense. Oh, I don't know why I'm obsessed with shirts, but a kind of a painting that can sense like react to the air or like biosensors that react to something and then produce a light in certain. Because I remember there was actually like an
Sure.
invention not too well, I know I say not too long ago. It was about a decade ago where someone like created some bacteria that would like express a certain dye after a certain time, so that people. So if something's been defrosted or refrosted, it can like
The color would
tell you
change.
whether like how much time it's been in that
No.
condition.
That's a good idea. I like
Yeah.
that.
So that was
Hmm.
that was an idea, but I
So again, like, and this would give you the scaffolding, right? Like for such a thing, like you wouldn't have to just mix bacteria into it. You could just maybe put a
yeah.
bead in, right? Like a bead, and that bead has the matrix, right? And it has the bacteria doing its thing.
Yeah, like, oh, yeah. There's a lot of like potential here, and there's a lot of like imagination. I think that bring like making more people aware of this technology and having people with the ability to create bioprinters can spark the imagination, and that could create lots of yeah,
Oh yeah, absolutely. It's a whole other realm of fabric, like material, right? Like once you have so much more work that goes into shape and like, oh, like how do you get that material to adopt different shapes? What shapes are good for what functions? And like, yeah,
I mean,
it's
even
exciting.
going back to like they show a
Have
pyramid like shape, and that reminds me of that paper we did a while back where like in the region of the biofilms, like the interior becomes quite anaerobic and the exterior becomes aerobic, and that changes the
yes.
way the organism's like physiology works. And I was thinking like, well, with this you can actually use
Yeah.
that as a method of control, and if you
Yeah, no, that's what I was saying. Like I'm imagining like a
yeah.
bubble tea, but like it's a bioreactor, right? But each of those bubbles are like they're the right environment for a specific organism that lives in there, right? So then you can have cross feeding between the bubbles and the
Yeah,
liquid.
I guess you could find it, make a less muddy kombucha, I guess, where you you just have like all the fungi in a
Oh, that's
bubble.
true. Funny shaped. Yeah. Yeah. That would be interesting.
Ah,
Okay. And then our last paper for today is also biofilm inspired. This one, though, is about microbiologically influenced corrosion inhibition of carbon steel via biomineralization induced by Shewanella putrefaciens. And this is essentially saying that you got a piece of steel and it gets covered with this biofilm of bacteria, and that bacteria deposits a protective layer of minerals that prevents it from rusting.
yeah, I love this. This has got like top image. We've got this paper this I think they call like
A coupon.
a
They call them
coupon
coupons. Yeah.
of metal, and they immerse it in water, and then it starts to rust, and you can see all these cracks happening, and then they immerse it with water with the Shewanella in it, and that one, instead of rusting, the Shewanella forms these like calcium deposits on top of it that almost produces like a pattern that protects the metal itself from being eroded. And that's a really cool technology, and they even talk about like self healing as well, where like if so, I'm gonna scroll down.
Yeah. I think,
They like
yeah, that one's in figure
yeah,
nine.
figure 9, where you if you scroll down to all the way
Figure nine, they have like they scratch the coupons, and then they show that over time in the Shewanella infused mixture, those scratches get filled in by the mineral
yeah,
deposits.
and that's like kind of an important thing. Sometimes you have like a pattern and they get scratched, and that can be an area of attack for like oxidation and for breaking down the material.
Totally. And I think what's interesting here is that there's also this related field of
Mm.
biofouling, right? Like it's also known that certain bacteria growing on a biofilm on metals can erode it
Mm.
faster. And so this, I think, opens up this. I made this probably people in this, right? But for me, this is my first sort of encounter with the idea that, oh, like it actually matters like what communities of bacteria we're able to cultivate on these surfaces, right? How do we encourage the bacteria we want to grow on certain surfaces, right? And discourage the ones that we don't. And it has these kind of wide-reaching applications because like this is like these are
Mm.
metal objects. This is like
Yeah.
boat hulls and airplanes and like right. Just imagine like a protective biocoating on all of these different things that right now I don't think we think of as being covered in at least not helpful
No.
microbiota. But even our inanimate friends can have helpful
Yeah, I mean,
microbiota.
apparently this study was in like inspired by archaeological finds where they'd found like some metal from like hundreds of years ago that was still preserved really well, and it was related to Shewanella,
Mm,
just being in the right place and the right time, and
yes.
having like the ability. I mean, if only there's some way to like make sure that we could have the right communities. I mean, I guess you could bioprint some stuff on the surface
Yeah.
of. You can bioprint Shewanella on the surface of metal.
Sure. I mean, I think already, like, if that thing is sticky, yeah, if that ink is sticky enough, then you get this thing covered in a sticky biofilm matrix. Um, But yeah, that's the whole paper is very engineering.
Yeah.
Actually, it's not from a microbiology journal. It's from a
Materials journal,
from a
right? Materials degradation.
yeah, it's
From a material.
yeah, it's from a materials journal. So, they just have their favorite bacteria and they're characterizing like what exactly is the minerals that are being deposited. I think they're
Well, I mean,
calcium
yeah.
deposit,
So,
a
yeah, I think
carbonate
it's like
deposit.
calcium mostly and calcium carbonate that deposits on the layer.
Yeah.
Yeah.
So a little salty.
So a little salty corrosion
And
resistance.
yeah, I mean, this is kind of like why I like microbiology because it has all these applications. It is like there's so much like diversity within it that you wouldn't really like
Yeah,
makes it very fascinating for me personally, and it's kind of why I enjoy doing this journal club.
totally. Yeah,
Of
no.
just like where you get to see all the different things that these tiny little organisms do to
Right.
yeah.
Right. And I think that's a great.
Right.
And this
And I think that's a great segue. If you have found something really cool about microbiology and you want us to cover it and like give it a read and have a discussion, then let us know. Join us next week for more microbiology
And we want to
news.
remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree.
I totally agree. You can reach out to us over Twitter at microtwjc, and we both believe that what we're doing here is peer review, and it's a process that we can all participate in. And so we hope that you've had a good time listening to us ramble on about microbiology today. If you have a correction or something you found unclear, just let us know.
It's
in
been
the
a pleasure
comments.
chatting with you, Danny.
Same here, Buzz. And
Same here, Fuzz, and thanks for the people who
Oh my gosh,
chatted
yeah,
in the
thank you
chat.
so much. This has been amazing to hear from everyone.
I've had a good discussion.
Yeah, we've had a good
Yeah,
discussion.
tune in next week for more microbiology content. Bye.